I'm going to present REP, a novel algorithm for motif finding from protein binding microarrays, and I will claim it is accurate and fast. Okay, so I'll start with a short biological introduction. As we know, gene expression is mostly regulated by transcription factors, which are proteins that bind to short DNA subsequences, which are termed binding sites. These are usually found in the promoter region. And by this binding, the gene transcription is either promoted or impeded. And the binding affinity affects the level of gene expression. So we are interested in modeling and learning the binding affinity and binding preference of those transcription factors. Now, in recent years, there were many technologies that were developed to measure TF binding in high-throughput manner. These include both in vivo binding, such as ChIP-seq, and in vitro, as protein binding microarrays. The most popular model to represent TF binding preference is the position weight matrix. In this case, in this matrix, each column contains a distribution of probabilities over the nucleotide, which represent the binding preference in that position. One of the properties of this matrix is that the positions are independent of each other. This has been shown to be untrue for some cases, but still this is a very useful model and it's commonly used. A convenient way to represent the matrix is using the logo format. So, a short introduction for protein binding microarrays. This is a technology from Bulick's lab. In this DNA microarray, there are around 41,000 DNA probes, each containing a unique sequence of 36 base pairs. These are designed to together cover all possible DNA tenomers. In one experiment, the TF binds to all those probe sequences, and the binding is measured using a fluorescence tag. And the final readout, the output, is the TF binding intensity to all the probe sequences. Now, the main goal from this data is to learn and infer the binding site motif. There were different methods already developed to find this binding site. These include seed and wobble, rank motif, Bimmel PBM, and Amadeus PBM. Two comparative studies compare different models and different algorithms that were designed to find and model the binding preference from protein binding microarrays. And one of the conclusions that they found that there is a division of the methods into two categories. One are methods that produce motifs which are degenerate and are very good at predicting in vitro binding, while on the other end, the other type of methods produce motifs which are similar to literature motifs and are more strict. In addition, both studies have shown that the PWM model is quite accurate for predicting high affinity in vitro binding. And unfortunately, they found that in vivo binding is much harder to predict than in vitro binding. And I think this illustration of the logos, they show this conclusion where the top logo is taken from JASPAR database. This is for the RAP1 yeast TF. And now the two methods, Amadeus PBM and seed and wobble, produce motifs which are quite strict. As you can see, they have the strong letters, and they are similar to the reference motif from JASPAR. On the other end, the two other methods, rank motif and Bimmel PBM, produce degenerate motifs, which are not so similar to the reference, but are better at predicting in vitro binding. So, following these conclusions, we raise the following question. First, can we develop a method that will be able to produce motifs that are both good in predicting in vitro binding and produces motifs that are similar to the literature motifs? Second, a natural conclusion that came from these studies is that in vitro binding is better predicted using degenerate motifs. And we ask, is it really the case that high degeneracy is required to predict binding well? Third, we were interested in learning about what should be the length of the binding site. Is it 8 positions, 10s, or more? So for this aim, we developed a new algorithm called REP, which stands for Rank, Align, and PWM. This is a simple heuristic algorithm that works in 4 phases. First phase, all 8-mers are considered, and they are scored according to the average binding intensity, meaning the average binding of all the probes they appear in. The top 500 are taken, and these are aligned to the top-ranking 8-mer. This is almost always the consensus sequence, meaning the 8-mer that the TF likes to bind to most. Then, using this alignment, we build a weighted position weight matrix. So we look at each column, and we deduce the different probabilities according to the scores given to the 8-mers of these nucleotides. And the last phase, we extend to the site positions by looking at the original probes in which the top-bound 8-mers appeared in. Now, due to efficient implementation and the fact that we work on such a small data, only top 500 8-mers, running time is only a couple of seconds. Here is an illustration of how the algorithm works. It's quite simple. So we have the top-ranking 8-mer, and then all other 8-mers are aligned to this top ranking. The weights are on the right side, and we can deduce the binding probabilities. To test and evaluate our method compared to the previous methods, we evaluated all methods in 3 different domains. First domain is the in vitro binding prediction. In the second domain, we checked the similarity to literature motifs. We tested how similar they are to motifs found by different technologies, not protein binding microarrays. And in the third domain, we tested for in vivo binding prediction. So in the first test of in vitro binding prediction, we downloaded 316 paired PBM experiments from UniProbe database. Since we have a pair of experiments for each TF, we can learn a position weight matrix from one array and test it on the other array, where we use the PWM to rank the probe sequences, and we compare this ranking to the original ranking according to the binding intensity measured in the experiment. We used 3 different criteria to gauge the performance. One is AUC, sensitivity, and Spearman rank coefficient. And in all criteria, our algorithm REP achieves the best performance. Here we show the average performance over these 316 tests. This is followed by BML PBM, rank motif, and then last is seed and wobble. In addition, we tested the similarity to literature motifs. We used the databases of CERTF and JASPAR, from which we downloaded 58 yeast and 59 mouse transcription factors that were learned from other technologies. And we measured the distance, the Euclidean distance, between the position weight matrix learned by each method to the one from the database. Again, we can see that our algorithm achieved lowest dissimilarity values. Here we show the averages. This is followed closely by seed and wobble. And rank motif and BML PBM both produce motifs that are less similar, and they have higher dissimilarity values. Now we were also interested in measuring the information content of each position weight matrix. The information content gives a sense of how degenerate the motif is. We use the 8 core positions to measure this information content. As you can see, we have 2 extremes. The seed and wobble produces the most strict motifs with the biggest, strongest letters, as opposed to BML PBM, which is the most degenerate. We can say that REP is somewhere in the middle. Now we wanted, since REP is best at in vitro binding prediction, we questioned the claim that a good binding prediction is achieved by very high degenerate motifs. So maybe the degeneracy is just a property of the algorithm, but not something that makes the model better at binding prediction. In addition, we tested the effect of motif length on in vitro binding prediction. In the first test, we took a fixed size of motif length. We kept the most informative positions. And as we can see, there is a general trend. The performance decreases as the motif length is shortened. So we concluded the site positions have a significant effect, and they should be kept. I think this supports what conclusion and results we have seen today from Gordon Stock. In addition, we tested another way to remove the site positions, and this is by removing the most degenerate site positions using information content cutoff. This has a bad effect on motifs of rank motif and BIML PBM, which are the more degenerate, so more positions are removed, and there's little effect on rep and seed and wobble. But in general, we think this cutoff is quite crude and should not be used. And our conclusion is that site positions should be kept and hopefully learned correctly. The last test that we performed was for the in vivo binding prediction. Here we used a very popular data set of Arbison et al. In this case, we used the 69 yeast chip-chip experiments, in which the TF was also available with a PBM experiment. We used the 2 PBM arrays to learn a position weight matrix, and we used this matrix to predict the binding of yeast promoter sequences. We ranked those promoter sequences and compared it to the ranking according to the original study. Now we can see from the results that they are inconclusive. It is unclear which method performs best, since in each criteria a different method has highest performance. But what is evident from the results is that in vivo binding performance is much worse than in vitro. So there is still a challenge in this case to solve the problem and to predict in vivo binding accurately. So to summarize the results of all the tests, in the domain of in vitro binding prediction, REP achieves best performance, followed by BIML PBM. In the case of similarity to known motifs, REP achieves best performance, followed closely by seed and wobble. And for the in vivo binding prediction, there is no clear winner. All methods perform much worse. Okay, so to summarize, I've presented a new algorithm, REP, which we say it achieves the best of both worlds, meaning it is both best at in vitro binding prediction and in similarity to literature motifs. REP is also extremely fast. We believe that the advanced performance is due to the concentration on the top 8 mers, which are the essence of the binding preference, but also the extensions to the site positions, which increase the performance. The tool is freely available on our group's website. In addition, we have additional conclusions. First is that high degeneracy is not required for accurate binding predictions, as we have seen that different degenerate motifs perform well in binding prediction. In addition, site positions help in predicting TF binding, so they should be kept and learned. And that in vivo binding prediction from protein binding microarrays is still difficult, and this remains a challenge to solve. And as we saw, many of the talks today try to tackle this problem. Okay, so I finish by thanking my colleague, Eran Mick, and my advisor, Professor Ron Shamir, and the different funds that helped us conduct the research. So thanks for your attention.
I'm okay to move on if we're out of time, but
No, we have time.
okay. So the question is, if you have several motifs, for example, or if you have non-PWM based models that truly correspond to the binding, the metrics that you're using of basically asking, okay, we're going to find a PWM that is, best matching known motifs, is a little overfitting to the fact that known motifs were discovered by PWMs in the first place, and also by this assumption that there's one motif that describes the binding. So I'm just wondering if, given your new algorithm, where in fact you have some hints that there may be additional motifs or that they may be
Well, we try to tackle this problem using our algorithm, but since we rank all 8 mers, even if there is a secondary motif, it's usually very close, or at least have some overlap with the consensus sequence. So we were unable to divide the 8-mers between those that belong to the secondary motif or the main motif. So for now, we left it out of the scope of this study, but these are excellent questions to solve. Yeah.
Thank you.
We have time for one more question.
Do you consider the sequences, the k-mers that are not enriched? Because I guess alignment is phylogeny-based, and that's because you cannot observe the species that don't exist. But here you actually see the k-mers that don't bind. And at least some of the problems in the earlier algorithms were based on the fact that they didn't consider the ones that did not bind. And they made basically a PWM that really doesn't explain the data, because you can see k-mers that have very bad scores.
So we used only the top 500 8-mers, and that was, enough for the algorithm. Usually the low-ranking 8-mers are more affected by a non-specific binding or background binding, which is affected by other factors, not just the specificity of the TF. I think.
Your model should explain the enrichment of the k-mers, and often in the previous ways of doing this, the PWM actually predicted that some k-mers that bind very poorly bind well. And this is simply because the alignment should consider the negative scores as well.
No. So we haven't taken this into...
Particularly with dimers, you get half sites, for example, instead of dimers and things like that, if you don't consider the poorly bound sequences.
So we can discuss it offline.
Yeah, you should
probably
consider it.